So we will start the viral DNA replication, and you will see that after these two intense chapters, you will feel that this chapter will be a little bit, not the last part, but some of the part will be much more easier, because this is similar to, as I said, prokaryotic and eukaryotic DNA replications are similar, so looks much similar. So here we are looking at these viruses. So we are going to talk about the small DNA viruses, single-stranded, like a parvovirus, and we're going to look at some of these hepatitis B virus, but that is a double-stranded DNA, but we are going to talk about that in somewhere else because of the complex process of that. And we are going to talk about the adeno herpes simplex and the papilloma viruses as well, like SV40, HSV1, adenovirus that we are already talking about. So that's basically that how they make their genome. Remember that last part we talked about that how the RNA is made or the mRNA is made and spliced and modified, and here they are making the genome. We talked about both RNA to RNA synthesis which in a genome as well as mRNA in the first topic of this slide, first topic of the module 2. Second one, we talked about the transcription. Here we are talking about the genome replication of the DNA viruses. So universal rule, this all of you know this. So I'm just quickly just going into that DNA synthesized in a template-directed incorporation of the deoxynucleotide monophosphates. The triphosphates are the building material, but when they are incorporated, two of these phosphates goes out as inorganic phosphate, PPIs, and the nucleotide monophosphate is in the chain. And always synthesized by a 5' to 3' semi-conservative replication to daughter cells. Replication initiates at specific site called the origins or ORI. We are going to use this term all the time, ORI, and ends at a terminus or multiple to plural termini. Now, again, remember that we talked about a lot RDRP, RNA-dependent RNA polymerase, which makes the mRNAs and the genomic RNAs for the RNA viruses. The second chapter that we just finished, for making the mRNA, we have DRP, DNA-dependent RNA polymerase, specifically RNA polymerase 2, makes the mRNAs. Now we are going back into the DDP, DNA-dependent DNA polymerase. So just make sure that you have the understanding of these, all three of these. So that's basically what it is. So primer dependent, and there are some rare cases of primer independent synthesis as well we are going to see. And basically from the parent strand, so you have the olive and the green if you just look at that. So this is one of the parent strand, and the pink and the a little bit darker pink are the newly synthesized. So always what you are seeing in the replicated DNA, you have one parental strand, one daughter strand, and that's why we call it semi-conservative. Now, conservative replication, does it happen? I just put a kind of a funny note at the bottom. It just discovered in 2013 that when you have a double-strand breaks in yeast, there is some conservative replication that happens. So this is for a question that you can trick a biology student to ask that and have that reference ready. And it's like, okay, conservative replication is the rule, or semi-conservative replication is the rule, or the conservative replication rule. I can guarantee 99.9% will not believe that there is a conservative, because no textbook will tell you that conservative DNA replication actually happens, but there is an exception. So that's just for fun. I put that reference at the bottom. But nothing to do with here as such. We are going to see all semi-conservative replication in the whole virus replication. So this is again a recap from the previous part. So what you are seeing right here, as you can see, that the only difference is that when we talked about these particular pictures in the RNA-dependent RNA polymerase, this was instead of T, this was UCG. So this is your RNA pol, or the pol 3D of the poliovirus that we have already seen. If this is doing the process of RNA synthesis from an RNA template, this will be EU for sure. And this will be RNA polymerase. Whereas this is a structure of the DNA polymerase where you can see that right here at the palm, this is right here is a palm of the hand, which is right here at the base. That's where the DNA is bound. That is the active site. And here you have the thumb and the fingers. Here you have the thumb and the fingers. So basically, the structures of, as I said, all polynucleotide polymerases, that means RNA polymerases, DNA polymerases, share a basic structure. And the same way that we have seen these reactions as well, that usually a divalent metal ion like a calcium, magnesium, or manganese, here the example of manganese, Mg2+, they coordinate these alpha and the beta phosphates for the nucleophilic attacks to add the bases one after another. It is the same mechanism that we have seen. So there is nothing new in here. So that's why it's very self-explanatory. So the only difference is that we have seen that there is at the active site, there is a different amino acid present which can accommodate the deoxynucleotide monophosphate, which are different from the nucleoside monophosphate. The ribose sugar does have a hydroxyl at the 2' end. Deoxyribonucleosides, which are put into the DNA at the 2' end, you do not have that hydrogen, you just have, you do not have the oxygen, you just do have a hydrogen in the 2' end. So that's basically what the basic difference is. We talked about that. So very general understanding, just only need some basic idea that viral DNA replication requires synthesis of at least one viral protein, sometimes many, and always delayed after the infection. Remember that that's the reason we talked about mRNA earlier, because that's the event that happens first. DNA replication happens later. That's why we talked about the transcription before replication. Some simple viruses requires more host protein, and if they have a smaller genome. Complex viruses encode many, but not all proteins required for the replication. Some of the host proteins are needed for sure. But interestingly, we are going to see that some of the DNA polymerases like pox viruses, which is the picture at the very bottom, the cartoon, the DNA polymerase is even produced by the virus, and the virus make this replication of the DNA happens in the cytoplasm, not in the nucleus. So that is an extreme case of large DNA viruses, but still they will need some cellular proteins for the help. They cannot do it all by themselves. So right here, small DNA viruses encode the entire replication system that we were talking about, encode the protein that orchestrate the host. What we mean by orchestration, that these proteins made by the virus channelize instead of the host DNA replication, they channelize their function to make the viral DNA replicate. That's basically hijacking the system. That means what we mean by orchestrating the host, making the protein do a different function, cellular protein do a different function for the viral replication. Large DNA viruses like herpes viridae adenoviruses make their own replication system. Most, remember that is most, sometimes not all. So that could be a difference in between your quiz question, that if you see that large DNA viruses encode all of their replication system, that will be a wrong statement. Most, not all. At least one or two proteins are needed for them as well. So what are the proteins required? You need the DNA polymerase. Of course, it is a DNA-dependent DNA polymerase, or DDRP, accessory protein. You need these origin binding proteins, helicases, to unwind the DNA. Exonucleases, these are needed to be, you are going to see that discontinuous synthesis happen, and these exonucleases are going to take out some of these residues from the overhangs. And also you need the thymidine kinase, ribonucleotide reductase, UTAPase. These are for the nucleotide metabolism that is basically needed. So here we are looking at the different viral origin of replication. Why it is important? Because this will be the start site from where the DNA replication begins. Our chromosomes have tens of thousands of origin, because if there is one origin of replication, one chromosome will take weeks to replicate. So there are many. So if it is a simple virus with a small genome, replication origin could be simple, but we are going to see that it will quickly change. So say SV40 parvovirus or adenovirus. So SV40 parvovirus, these are two small genomes. You have one origin here, one origin here. Adenovirus, you have an origin at both ends. Herpes virus, you have an origin right in the middle here, and there are two origins right here. And there are many terms in here, and we're going to look at these terms. And say for an example, for adenoviruses, this ITR is an inverted terminal repeats, and the TP is a terminal protein covalently attached to the 5' end of the genome. Parvovirus, you have this T-shaped structure at the end, and these are the terminal repeats, and they are in palindromes. We are going to see that they read forward and backward the same way, which gives this palindromic structure. And this rep-ORF is the replicase ORF, or this makes the replication protein, related proteins, and the cap-ORF is the capsid protein. So replication related proteins and the capsid protein. And these are the transcription site. Herpes virus, what you are seeing, that you have a long stretch, we call this as unique long region and a unique small region. At the end, you have a terminal repeat long. At this end, you have a terminal repeat small. And in the middle, you have an internal repeat long, internal repeat small, and that's what it is. So, and terminal repeat small. And you have an origin S at both ends of the small segment and an origin L at the very middle of the large segment. So you can see that multiple origin and much more complex. So that's where we will stop, and we will start from the key features of the viral DNA replication in the next slide, next class. Yep. So we are going to start from this slide, and we were talking about the DNA replication. And what you are seeing basically in the slide, there are just 3 examples of the origins of the DNA replication for SV40, HSV1, and adenovirus 5. And the first question that you will probably ask here is that what I am supposed to know from this slide. Do I need to memorize all of them? No, that's not the purpose. So just take some key features, that's what it is. And you will see that the narrative is pretty long, but there are some take-home messages. So the first thing what you are looking at, that there are some core origins. The yellow sites are right here, sequence bound by origin recognition proteins right here. So yellow sites are those. That's number 1. Then when you are looking at those sequence, they do have some very specific specific characters. What we mean by that, if you're just looking at these arrows, like these black arrows, those are repeat sequences. If they are facing the same way, they're tandem repeats. You have a repeat, ABCD, then ABCD. If you see that they are backwards, that means they are a palindrome. That's right here, you have an ABCD. Here it will read CB, for DCBA. If you read it from this side, it will be ABCD. So these are the palindromes. You can see that. And on overall, this whole sequence, you can see that this is a palindrome as well. And these are some of the features of that. Here you don't see that. Along with that, what you are seeing, that they are close to some AT-rich element. Here's an AT-rich element, AT-rich element, AT-rich element. And along with that, what you are seeing, that right here, you can see that this is the transcription regulator site. So the next part, what you are seeing, so just remember that this arrow, red arrow, means that the transcription starts from here. So many of these origin binding region is close to your transcriptional regulatory element as well. So as we said, they could be tandem or inverted repeats. They are close to AT-rich regions, and they are close to the transcriptional regulators. This is basically the take-home message. And what we are going to see, that just giving one example here, we talked about this LT, or the we're constantly talking about that, the large T antigen. The large T antigen is an origin binding protein, and that binds right here. One molecule, another binding binds here. And why two, we're going to see that. And there could be some imperfect palindromes and those are some minor things. But these are some of the take-home message. That's what it is. So that's basically what it was. So basically, on the slide, what you have, this is your take-home message. So after that, we are looking at some of the origin recognition proteins. This is just a description. The name of the protein is Rep7868 in the parvovirus, very small genome. And the name is the 78 kilodalton and a 68 kilodalton protein. That's why replication protein 78 kDa, 68 kDa. That's why it's called 7868. For the SV40, it's not written here, but you know that is LT. And that is LT, or the large T antigen. For human adenovirus, we are going to see that there will be some pre-terminal proteins. You do not have to write pre-terminal proteins along with some of the polymerases. So right here, what you are looking at, that there are some other proteins which are already bound. These are the terminal proteins, which is a part of the genome. That is not that is recruited. That comes from as the genome. Whereas this right here, viral replication proteins, is where a pre-terminal protein is bound with some other. Here also, you can see that the herpes simplex virus, there are many others. That's why names are not required. But you can see that it can bind to ORI-L, as well as there will be some replication proteins bound to the ORI-S, and there are multiple sites in there. And in the vaccinia virus, the replication starts close to one of these loop structure. Remember that these terminal loops do have these inverted repeats, and the two strands, the 3' and the 5' strands, are joined covalently end to end. Remember that the base pairs are different. This is like a 3' end hydroxyl is connected to the 5' end, so it is covalently joined, so it's a closed loop. Just think about that. Let's see that this is a single-stranded parvovirus. If I extend this strand where you have the protein and join it to this end, that will be pretty much it. That's it. So on the other hand, if you just look at this one, they are not joined end to end. These are not joined end to end. Of course, this is a circular DNA. This is also covalently joined end to end. So these are some of the architectures of that, and we will just go one at a time. So the large T antigen, or LT, of the SV40 is one of the most studied protein in any system, probably. Because this protein is so unique that it does it all. And just we'll take a look at that, what it does. So just starting from this end. So right here, this sequence near here, this is responsible for host range. It's a monkey virus. It's just infect monkeys, but there could be different other things that it can infect. And the species differences are dictated by that. Then what you are seeing, that this large domain right here, as well as right here, this binds to the Pol alpha, and this DNA Pol alpha is the primase which puts down the RNA primer during the replication, so we are going to come back to that. So it binds to another protein. Also, along with that, it binds to P53. And again, I mean, like, if you're not familiar with the P53, P53 is basically called the guardian of the genome protein. So it actually keeps the health of the DNA. This is in the host proteins, all cells, in a perfect shape. If there is a DNA breakage, it just brings up a lot of different protein to repair them. If they cannot repair that one, it just sends the cell to apoptosis so that they get killed, basically. So what it does, that the P53, it binds to P53 and sequesters it. That means that it inhibits the P53 function, because the virus is going to mess up with the DNA in the cell, and the cell cannot go into the apoptosis even if there is something wrong. So that's what it is. Then this region is the origin DNA binding domain that we talked about, that yellow sequences right here. We saw that. You can see LT binding site 1 and 2 right here, LT binding site 1 and 2, that binds to the origin. Then this overlapping region, one portion is a helicase. That means that will basically bind to the DNA and unwind it, helicase function. Also have an ATPase function. To do that process, it's an energy-dependent process. You have an ATPase function overlapping with that. and this portion will also bind to the ssDNA binding. As the double strand of DNA will separate it out, it will bind to the single stranded to keep it stabilized until there is another protein we are going to see is going to cover them all. The SSBs, or the single strand binding proteins, are going to cover them all to keep them stabilized so that they don't get annealed. Right here, this famous RB binding site. So again, remember that RB is also a tumor suppressor. So it doesn't allow a cell into going into the replication. But all the viruses wants the cell to replicate so that they do have enough replication enzymes, DNA polymerases, RNA polymerases. They want the cell to divide and make this protein by transcription. But RB checks that. When it binds to the RB, it abrogates the RB function. It doesn't allow the RB to repress transcription of the cell. And that helps the cell to make these proteins, which is basically exploited by the virus. Then the J domain is also, we are going to talk about that, what exactly what it is. We're going to come back to that. Also, what you have, excuse me, this HSC70, this is a heat shock protein, and this is basically heat shock protein 70 kilodalton, and this is a chaperone protein. It helps some other proteins to properly fold. That's the function. Then what you're seeing, that there are several phosphorylated sites, and most of them are serine, S-serine, and there is a threonine 124. So just out of that, just note that threonine 124, because we are going to see that the phosphorylation of this particular protein, which is a threonine 124, actually enhance the function or make this protein bind the RB. So the phosphorylation of this threonine is required for this protein to bind to RB. That's one of the most important thing. All right. So those are some of the functions of the T antigen. So this is what we talked about. Most of the cells do not divide so rarely, do not replicate the virus in the quiescent cells, and the virus must induce the host replication protein, and these are encoded by the viral early gene product. So they move them into the replication or the cell cycle in the cells for their own aid so that they have the replication proteins in there. So this is what we've talked about, that remember that the RB protein basically checks the cells to go from G1 to synthesis phase, which is the replication of DNA happens. So basically all the proteins DNA replication proteins start to synthesizing after that restriction point is passed in G1, and RB arrests them in there. And that's basically the overview of that. And we are going to see that, as we saw that in the LT, the protein, that when it binds to the RB, it cannot do the transcription. So this is, again, we are revisiting the same slide that we saw in the transcription. So this is RB, and it binds with the dimerization partner. We have seen it's not detailed, but in the last part, we have seen that the other part is the basically E2F and the DP. This E2F and DP binds into some of the specific region of the transcriptional control region. When RB is bound to that, it basically recruits the HDACs, or the histone deacetylases, and the DNA is tightly wrapped around. It's not going to do any transcription. All these proteins, SV40ALT, we have seen all these proteins, adenovirus E1A, they binds RB, and that's the reason that RB cannot be recruited in there. And if they cannot be recruited, then there is transcription. When RB sits in there, there is no transcription. In the cell, how it is controlled, RB, if it is phosphorylated by a cellular kinase complex, which is a cyclin 2 CDK2 cyclin A complex, again, that's not needed, but the cyclin 2 CDK complex that basically phosphorylate the RB in a normal cell. When RB is phosphorylated, it falls off and the transcription go on. And what we are going to see that this right here, this threonine 124 is phosphorylated by the same cellular kinase which phosphorylates the RB. So that's your CDK2 cyclin A. But what this does, that this makes the LT competent in binding the RB. So LT is now the SV40 is ready to bind to LT, and when it binds to the LT, it just cannot be recruited in here, and the transcription will proceed. Now, why we are talking about transcription in a DNA replication? Remember that what we are talking here, the transcription basically make the proteins or the enzymes for DNA replication. These are the DNA polymerases and all their subunit, and subsequently RNA polymerases. So that's the implication, that the DNA replication proteins transcription is enhanced by binding or abrogating these tumor repressor proteins like RB. All right, after that we are going into the mechanism of the synthesis, and this is old material. You have already seen this many places, so we will just quickly go over that. So a normal replication like these viruses, they just do the classical synthesis. You have a 5' 3' end in one of the leading strand. The synthesis is continuous. If the polymerase, basically the primase part, lays down the primer, and the DNA is continually synthesized as the replication fork is unwound. On the other hand, you have a 5' 3' problem, so it has to be synthesized because this is 3' to 5'. Always the polymerases work from 5' to 3' end. So in this strand, it has to be discontinuous, and this is once by a primer, then synthesis, primer, then synthesis. These are the Okazaki fragments, and then finally the RNA primers from the Okazaki fragments, also from here, are removed by some of the RNases, and these are filled by the DNA polymerase and sealed by ligases so that they will have the two strands. This is the normal synthesis. On the other hand, what we are looking at, that in the for adenovirus, parvo, or the pox viruses, there is a synthesis which we call as a strand displacement synthesis. You have a primer, and one of the strand is synthesized where the other one is displaced. So what happens to this strand? We are going to see that how this strand is going to be replicated as well. So this, you know, that you just going to synthesize another double strand, and that double strand is going to be replicated. So always you are making this strand, this blue strand, separate out, and how this is replicated by different strategies that we are going to see. But this is basically the strand primer dependent strand displacement method. So this is one of the easy ones, which is the replication bubble that is formed in a circular, covalently circular DNA. So these are from early electron microscopy, that in the origin, all the proteins bind and a replication fork or a replication bubble is made, which has two replication forks at this end and this end. And you can easily see that the synthesis will happen just like this in both ends until they go all the way around, and you will have a full double-stranded copy of the original DNA. And then these two are interlocked. So think about that these are interlocked, and they have to be cut out to separate. And we are going to see that, but we use a word that how these interlocked covalent molecules, double-stranded DNA molecules, are separated out. We call that as a terminal resolution. This is a very important word. Wherever it comes to as a terminal resolution of DNA replication, that means the newly synthesized double strand, how it is separated out from the interlocked mother strand, basically. Or mother, not strand, this is semi-conservative. So half of the strand is new, half of the strand is old, but the two strands are separated out. That's your terminal resolution. So here is just the, we saw this right here, and this is just happening in both ends. So here you can see that the DNA, this is the origin of the replication. This is 5' to 3'. This is the origin, so the synthesis is going on this, this direction. And then what we are looking at, that the synthesis on this strand, because when it is being unbound, this will go on as the lagging strand. So that's basically what the synthesis is. And on this strand, you can easily see that when the replication is happening right here, this is from the 5' to 3'. this is being made in a continuous form, and these are made into the discontinuous form with the Okazaki fragment as they are. Now, just make sure that you clearly understand how this is happening. Sometimes you just feel like, okay, this is 3' and why this is going in this direction and how it is 5' to 3', how this is happening. Remember that this whole strand is in the direction of the 5' to 3'. So when it is going in this direction, it is the 3', it's going into the 5' to 3' direction. So just remember that. So these 3' ends do not get confused with these ends. So take a look at that. But when you are doing this, there is a problem always that we called as in DNA replication the end problem. At the very end, if you have a double-stranded DNA, you put down a primer, and that primer will be removed. And how you basically make that make up that gap, that's the problem. Here, there is no problem with that because they are covalently joined end to end, and they can just make this. They will go around, so it's just like round, and it will go around all the way towards the end, and there is no end problem because this is a circular DNA. There is really no end in a circle, so it will go all the way to the end. So there is no end problem. So this is what we call as an end problem. See that these are the discontinuous Okazaki fragments, and elongated and excised primers, elongate, ligate, there is no problem. Problem is at the end. When, and we call this now what? But if it is a double-stranded, it's joined end to end, what will happen? That this will extend all the way to this end. Remember that. This will be extended all the way to the end because they are joined in a circular form. There is no end problem with that. So that's how it is solved when it is. And we're going to see some really interesting method when it is linear double-stranded. So in eukaryotes, the telomeres actually basically do that, the telomeres. So now what we are going to look at is the SV40 DNA replication. It looks a lot of things, but just see that this is regular DNA replication just with an addition of a viral protein and how it happens. Here you have an LT binding site, and here the LT is recruited. LT is right here is a monomeric form, and it is phosphorylated. And remember that it's an ATPase as well. When it is phosphorylated, it just basically forms a hexamer on both sides of the LT binding site. So it forms a barrel-like hexamer which completely surrounds the double-stranded DNA. And there are conformational changes in the DNA, and it recruits a couple of different enzymes, which is an RPA and topoisomerase 1. Now remember that, this is very carefully that what are the specific functions. LT, we saw from that previous slide, has a helicase activity. That means LT will unwind the DNA. When it is unwinding the DNA, there will be a tension that is formed because, you know, I mean, like just the overview of that when you're just moving the fork, it will form a tension. So that tension is released by what is, it cuts, loops the DNA, and seals. This is done by topoisomerase 1 right here. You are seeing that topoisomerase 1. This is doing that, is showing that. Now, the single-stranded DNA that is being formed by the fork, the RPA is a single-stranded binding protein that basically coats the protein. So that's the pre-initiation complex before that the material can happen. And here we are going to look at the origin binding protein in the last one. Is like you have the DNA, and we are going to see this is a clamp protein, PCNA, and the other factors which are associated with that. So these are all crystallographic studies that we are going to see. Now what happens? Once you have this single-stranded binding condition and the two forks are formed, these are the cellular enzymes. So remember that. All of these are cellular enzymes. Only LT is viral, RPA, topoisomerase 1, these are all cellular enzymes. And it's going to put the DNA polymerase alpha. So the DNA polymerase alpha, or the Pol alpha, is the primase which basically puts down the RNA primer. And of course, you will need the DNTPs for the DNA replication and NTPs. Remember that people is like, why you need the NTPs? NTPs are needed for what? Anyone? So it's a DNA replication, but you need the NTPs as well. It's very easy. Well, for the RNA primers, that's right. You have to put the RNA primers, so that you need the RNA primers for that. So once these RNA primers are put down, that's what you are seeing right here, this is in the red, then you put a couple of different proteins. One is called an RFC and pCNA. So the RFC is a clamp loader protein, and the pCNA is a clamp protein. What it does, that now they are going to recruit two different types of enzymes. One is, we're going to see that DNA polymerase epsilon, another is DNA polymerase delta. And these proteins are much more processive. That means that they are just not going to fall off, and that is basically the function of these clamp loader proteins, to make them processive, that we used all the time. So you can see that right here, RFC and pCNA with the help of ATP. First, this Pol epsilon is basically recruited, and Pol epsilon will basically make the continuous strand. Remember that Pol epsilon is responsible for making the continuous strand in the leading strand. That's basically the Pol epsilon is doing. And then, synthesis of long DNA. Remember that. That's the thing. Pol epsilon is doing that. The next, what it does, that you have already the RFC pCNA, and it recruits the Pol delta. The Pol delta is synthesizing the lagging strand or the Okazaki fragments. Pol epsilon, this continuous synthesis strand, is done by that. Pol delta does the other. So are these two entirely different enzymes? Absolutely not. They just vary in a couple of subunits. That's what it is. So the holoenzyme just vary that how it is. So that's basically what you are seeing, that these are the two leading strands, and this is right here is the lagging strand. And so right here you can see that the primers and the lagging strand. And this is basically the synthesis. The bubble extends all the way towards the end. And although in the picture it's shown that, okay, all right, you have the polymer, you have the synthesis of the Okazaki fragment like this, then that, but the polymerase actually don't move along that, but they are close to LD. So you can see right here, you have the Pol epsilon with the RFC pCNA processivity factors, and it's continuous synthesis, 5' to 3' end. There is no problem with that. As it is unwinding, it is synthesizing the strand. On the other hand, see that your Pol delta is also, along with that, is in the close proximity right here. And what it does right here is the Pol delta. And remember that your Pol alpha primase is constantly needed, because it needs to synthesize the primers constantly for each Okazaki fragments. And for the lagging strand, the DNA is actually spooled through, making a loop. It is spooled through the DNA Pol alpha and the clamps, basically. So they are sitting at the same position. It is very sometimes difficult to see that. So look at your supplemental video. There is a video animation for this, that how it is done. If you look at the animation one time, you will get it. So it's very easy. So definitely for this reason, you look at those supplementary material for DNA replication. All right, so here we are talking about that the terminal resolution and during the replication. So during the replication, what happens? We only in the previous slides, right? If you see right here, is only topoisomerase 1 is shown in this picture. But remember that the tension during the replication can be cut out by topoisomerase 1 or 2. It's 1 or 2, and one strand is cleaved, and it relaxes the supercoil. After the two strands are synthesized, the terminal resolution, this is your terminal resolution, this is achieved only by topoisomerase 2. So 1 and 2 during the replication, only 2 for terminal resolution. So you do not have any end problem because these are circular DNA. So let's now take a look at the parvovirus. Very unique virus. You have ssDNA. Remember, the single-stranded DNA, we do not see minus strand ssDNA or plus strand DNA. We cannot tell that because you can have basically the plus or minus is basically the same the way you are looking at it. For the RNA, we can tell that because it has to go, if the RNA is immediately making mRNA or ready for translation, we know it is plus. If it has to make a plus from the minus, we know that it is a minus DNA. In the DNA, you do not have that issue. So we usually always call it ssDNA, but it could be plus or minus, no matter what. You can call them each. So the parvovirus DNA does have this replication ORF. Replication proteins are made, and these are the replication proteins, P5, 19, and 40, transcription sites. Then this makes the capsid protein, just one capsid protein. It's just a T equal to 1. So you have 60 copies of the capsid protein in here. Now, here you have the terminal repeats. And these terminal repeats basically make this T-shaped structure. And here you have the origin with a 3' open end. And let's take a look at that, how it is. And if you just curious that how these repeats are, if you just read it from this T, it is C, G, G, G. And here you can see that it is G, C, G, G. So that's basically that how you are going to see that how the repeats actually basically work. So the picture is a little small. So I apologize for that because there's no way I have to just then separate it out. So let's take a look at how the first hairpin is actually formed, or these terminal loops are basically formed. So if you look at the 3' end, which has the inverted terminal repeats, you have the sequence like A, B, C, A prime, D. So A and A prime, these are the inverted terminal repeats. Then here on the other end, you have a D prime, A, C prime, B prime, A prime. So here you have A prime and A. So why this is formed? Because look at this, like I'm just looking at this end. Remember that this will loop out. You have an A, B, C, A prime. So A prime is complementary, and then D. On this side, you have A prime, B prime, C prime, A prime. These are complementary, and this is D prime. So this basically makes these hairpin-like structures, or the T-shaped structures at the not really hairpin, T-shaped structures at the ends. So let's see that how basically it happens. So right here, you have a 3' hydroxyl open right here. And right from this primer, so this is a DNA primer, no RNA. The DNA polymerase, basically DNA pol alpha. There is no DNA pol alpha, because remember that the inverted terminal repeat is a self prime, and it requires pol delta or FCPCNA. They would straight form all the way up and complete that strand. So this end is closed, this end is open. Now the interesting part happened. There is a replication protein we talked about. This is your Rep 7868. It makes a nick in between this A prime and D right here. It makes a nick right here where you're seeing. When it makes a nick, it is easy. You have this end is a 5' to 3' end, and from here, the RNA polymerase synthesize the rest of the strand. Now you have two double strands. But all of them, or both of them, do have these inverted terminal repeats at the end. So they are going to form a fork or the T-shaped structure at both ends. And let's take a look at how this basically happens. You have just right here, first we are looking at the red, the blue one right here, red and the blue one, the bottom one we are looking at. The bottom one will synthesize the same way that from this D prime, it's going to synthesize all the way to the end, just like this. It is the same way. The top one is this. The top one is going to synthesize like this. So you can see that right here, you have the Rep A protein at the end. So all the way towards the end right here, from this end, this will act as a primer right here. And remember that this is your 5' to 3' strand, and the hydroxyl is open at this end. And these 3' hydroxyl, the 5' end is going to extend all the way to this. And of course, this replication protein is removed. And that's it. And then from here, it just goes back to the original place. So, or the original replication that what we have basically. So these are the two ways that the two different strands are synthesized. There is no end problem because of the nick or the Rep 7868 nicks it and seals the end. And once it, not seals the end, makes the end, and that's it. That solves the end problem for parvovirus. Strand displacement. All right, so after that, we are looking at the adenovirus, and this is a protein priming. And the origins are at both ends, and this is also strand displacement synthesis. So we have seen for SV40, it is a closed circular, covalently closed circular. Then we just saw that it's a single-stranded DNA. Now we are coming into a double-stranded DNA and origin at both ends. And this also is semi-conservative, just like all other. So here, how it happens, that at the 5', one of the end, you can see the 5' has an attached terminal protein. And at the 3' end, where you will start the priming, you have a pre-terminal protein. Remember very carefully, a pre-terminal protein is recruited. These are viral proteins, which binds to the Pol alpha or recruits the Pol alpha. But this is the interesting part. This PTP does have a serine residue. And this serine residue binds to a cytosine covalently. It's a covalent binding of a cytosine, C, right here. And always the 3' terminal right here of the virus is a G. So basically what you will see that this is GP means like it's a nucleotide DNMP, monophosphates. So you have basically the sequence is G, T, A, G, T. That's what it is. But the G, the first one is the most important one. You have a G and it base pairs with a C with a 3' hydroxyl open. So the DNA polymerase will start right from here adding so if it's a T, it will be an A, T, G, C, T, A, and it will go on. That's it. So this is your primer, so protein primer. So again, it's a strand displacement. It just recruits right here, and the strand displacement synthesis. This is the primer, and the polymerase is going to go and displace this strand as it goes on. It synthesizes the new strand in the red. As the other strand is displaced, it is coated immediately by another protein, which is called a DBP or the DNA binding protein. And this DNA binding protein basically circularizes, because one of the most important part that you can see in the last one, that each one of these strands have an inverted terminal repeats at the end. You have an ITR here, you have an ITR here. These are the sequences that will basically complement each other because they're inverted repeats. So what happens? The ends, basically A prime, they are complementary, and it will circularize this molecule, the ones which is displaced. So let's finish the first one. So you can see that this is going all the way towards the end, and you have a double-stranded molecule right here, which will again start synthesizing a new strand. No problem with that. How the other strand is synthesized, just remember that you have this completely circularized molecule. The interesting part is right here, that this is, if you carefully look at that, this is your 5' to 3' end. So you have a 5' to 3' end, and there is absolutely no problem. You have a pre-terminal protein the same way like this, and it will start with a serine and a C, and this strand will be synthesized all the way towards the end. And as the strand will synthesize, this single-stranded binding protein will be displaced. And then you get back basically this. That's basically what it is. So this is how the other strand is replicated. And then your question could be, what happens to this pre-terminal protein which is attached to that? This pre-terminal protein has two protease cleavage sites. So there's a viral protease that cleaves it, and it remains there as the terminal protein. These are all written in your material. So the pre-terminal protein is processed by proteolytically to the terminal protein. So that's your adenovirus synthesis. All right, so this is just a molecular structure that how the adenoviral single-stranded binding protein, which is a DBP, is incorporated. There is really nothing. This is a plow-shaped structure that is the molecular model that how it is staggered on top of each other. That's basically what has been shown. I mean, like really nothing is required as such information-wise, just then or the idea. Then we are going into the herpes simplex virus, and it is extremely complex. And you can see that I wrote all these names, but I will never ask you that what is the function of UL29, UL30. But you can see that there are a lot of these functions for these, and it codes its DNA polymerase as well. Not all of it, but part of it is produced by the virus as well. Large virus, 150 kilobase genome, whereas adenovirus a little bit smaller, is about 35 kilobase, but this is like 3 times, 4 times the size of an adenovirus, very large virus. And the DNA basically is converted into a circular molecule, and it replicates as a rolling circle. So it's a very interesting idea. So you may ask that why I just put all these names in here. We are going to use some of these names when we come to the packaging of the genome. That's why you can just read it, and if it's in the back of your mind, that's good enough. But no direct question about memorizing. You may have a quiz question because it's open book, but not in the exam. All right, so first what happens, that as soon as this linear DNA enters into the cell, delivered into the nucleus, it is circularized by a DNA ligase 4 or an XRCC force. So XRCC4, now remember that there is a very close something like tongue twister like that. CXCR4 is a co-receptor for HIV to entry. XRCC4 is different entirely. So it is X-ray cross complement. So if you have a DNA breakage due to the X-ray or irradiation, this is the protein that repairs or closes those DNA. So it's a DNA ligase. So it is circularized. So these are host proteins, remember that. So these two are kind of important host proteins responsible for circularization. Now what happens, that right here, there is a nick that is done by an endonuclease. Right here, an endonuclease, remember that endonuclease always cuts in between, exonuclease from the end, endonuclease can cut in the middle of the DNA. And you have a nick, and a 3' hydroxyl opens up. And you have this DNA is being synthesized by the DNA synthesis machinery, like this goes on. But what will happen that once it is synthesized up to this, you have a problem. There is not much space for this to go, because this is like a replication fork. So basically what happens, the DNA synthesis is discontinuous by forming a primer. And again, as you can see, the green part is the RNA primer done by the RNA primase, and then you have the DNA is synthesized with that. And then the RNA primers will be removed by the RNase H. And this is one of the things that I also probably should have mentioned. It's written in there, but I did not do that. So right here in the SV40, you can see that RNase H basically removes the primer from the RNA-DNA duplex, and the FEN1 basically cleans out these 5' overhangs, and the DNA ligase will seal the gap. Of course, the gap that is being formed will be repaired by the DNA poly delta or epsilon. So, and the DNA ligase will seal the gap. This is the same thing that is going to happen in here as well, that these primers are going to be removed by the RNase H. If there is an overhang that's cleaned up by the FEN1, and the DNA polymerase is going to fill those gaps, and they are sealed by the DNA ligase. And all these genomes are end-to-end joined, and this forms a concatenameric DNA. It is end to end. And that's the reason that there is no end problem to that, because it is just joined all the way towards the end to end, as you can see. So what we are going to see that these concatenameric DNA, which is attached end to end, they are going to be excised out for packaging into the genome, into the viral particles. That's the next chapter. But that is the reason that you do not have any end problem in the rolling circle replication. So here, the pox virus, and again, you can be a little bit intimidated by why you have this slide even here. This is just to show, you do not have to memorize a single name of the protein in here, just to show that the pox virus basically make every protein that is required for its own replication. So the take-home message, it doesn't replicate in the nucleus. It sets up its viral factory to replicate in the cytoplasm. Remember that. Herpes virus, adenovirus, all of them need to go to the nucleus because it exploits the host DNA polymerases and the machinery for the DNA replication. Here you can see that all of the proteins that is needed, including RNA polymerases, they set up the factory in the cytoplasm. That's basically the only take-home message from here. And this is some experiment. So you have these cells. This is stained with a DAPI. DAPI is a DNA stain. Wherever you have stains in the cell, they are going to be stained with a DAPI. And here you have a very specific DNA binding protein, a specific stain for the pox virus. And you can see that right here. This is the pox virus, A5CFP and YFP attached with those. And these actually light up right here. This middle portion is the nucleus. This is outside the nucleus in the cytoplasm. So you can see that these are the viral factories which are in the cytoplasm, not in the nucleus. So that's the experimental proof of the DNA factories in the cytoplasm, that how they do this. Now, the pox virus DNA replication is still a kind of an enigma that is not fully solved. There is two schools of thought that basically think two different ways. So the DNA is, remember that, it is like this, circular, or we can think about that as linear, but the ends are covalently joined. So that's basically what it is. So one group of scientists basically thinks that it just makes a nick in one of those and extends that, extend that strand. That is basically one of the syntheses. Another group of scientists basically think that there is a discontinuous synthesis by a primer, and that's basically what it is. And as it is end-to-end joined, just like a circular DNA end-to-end joined, there is no end problem. You can easily see that they are basically end-to-end joined. Here, if you just look at the structure right here, see that you have the inverted terminal repeats at the end, and they are covalently joined right here, and you do not have any end problem with that, no matter that how it is. And then, of course, what happens that the topoisomerases, that type of protein, they just remove these concatamers, basically. That's what it is. So when you are looking at this structure, genome monomer, and this is VACV, means that is a variola virus, a pox virus. A common model that we use is a variola. Remember that a pox virus, your smallpox virus is a pox virus. Chickenpox virus is what? Anyone? Is it a pox virus? No? Anyone? It is not. It is varicella zoster is a herpes virus. Remember that. It is a herpes virus. Your chickenpox virus is a herpes virus, varicella zoster. Your smallpox virus is this, and vaccinia and variola, these are the pox viruses. All right.